Hey there, everyone, and welcome to this video lecture for Psych 370. In this one, I'm going to be going over the different sources of data that are described on pages 42 through 49 of the textbook. So I'll be talking about some of the more common methods that learning researchers use and basically how they collect their data on learning and behavior. Now, probably the worst way for a learning researcher to collect that data is to rely on what we call anecdotal evidence, or sometimes we call it hearsay. So, anecdotes are notoriously unreliable as a way of knowing about things. But having said that, we encounter anecdotal evidence all the time. It's very common. So, an anecdote can be defined as an account of something that's based on personal experience with it. Okay. So, for example, some people believe that dreams can sort of foreshadow future events because they've had experiences or they've heard stories where something that happened in a dream actually occurred in real life a few days later. Now, of course, there's very little scientific evidence that dreams can predict the future like that, but some people nevertheless believe in this sort of prophetic quality of dreams because of anecdotes they've heard to that effect. And as for all those cases where what happened in a dream didn't happen in real life, or where what happened in the dream was different from what happened in real life, well, people who rely on anecdotal evidence for their claims tend not to notice or remember anecdotes like those that run contrary to what they believe. Or here's another example like that. It is a widespread belief among people in general that sugar causes children to become hyperactive, right? Lots of people think that's true, probably because they remember a time or two where a child did have some sugar and then got really hyper, or they've heard about other anecdotes like that from other people, like, say, a parent who says, "Hey, watch out how much birthday cake you let my little boy have because, too much sugar," and he just gets out of control. So, anecdotes like those are pretty common, but scientific research indicates that that idea about sugar and hyperactivity is really just a myth. Sugar doesn't actually cause children to become hyperactive. So, because of the selective, unsystematic nature by which anecdotal evidence tends to be collected, it is clearly flawed as a way of knowing. It's just not a great way to get to the actual truth of something. But one nice thing about anecdotal evidence is that it can inspire hypotheses that we can go out and test using other research methods. So, for example, hearing those anecdotes about sugar making kids hyper could have been what led curious researchers to conduct controlled experiments in order to determine whether that common wisdom was actually supported by the scientific evidence. So, anyway, anecdotes represent one source of data for people who study learning. And another way to collect data is to conduct a case study, which, as the name suggests, is a detailed, in depth examination of a single case, such as a particular individual. Now, case studies are especially common in medical research. Okay, so for example, we can get some insight about the possible functions of a particular brain region by conducting case studies on individuals who have suffered damage to that brain region. For example, one such case that you might be familiar with is that of a man named Phineas Gage, who in the mid 1800s experienced massive frontal lobe damage when an explosion caused an iron rod to go shooting through his skull. And since Gage's personality reportedly changed after this, because he apparently became a much more impulsive person after the accident than he had been before, his case suggests that the frontal lobes play an important role in personality and impulse control. So, people with damage to a certain brain area often make for interesting case study subjects. Other times, a case study might focus on someone who's highly gifted in some way, with the idea being that by examining this person's life in greater detail, you might get a better understanding or at least a possible explanation of how exceptional skills like that can be acquired. Sometimes a person with a rare disorder might be the subject of a case study. So, again, in a case study, you're doing this detailed examination of a particular individual, their behavior, their condition, their upbringing, their family background, and so on. Anything you think is interesting or important. basically. That's what you'd include in a case study. And case studies do represent a better way of collecting data than anecdotes simply because your observations are being made in a more structured, more systematic way. But case studies do also have a couple of limitations that are important to acknowledge. One is that it's very problematic to try to generalize from what you find in a case study to a larger group of people or a larger group of animals. Because after all, when you conduct a case study, you're dealing with a sample size of one, right? And it's virtually impossible for a sample that small to be representative of the larger population to which the individual belongs. So, generalization is difficult. And another limitation of case studies is that they don't allow us to draw conclusions about cause and effect relationships. So, for example, the case of Phineas Gage may suggest that the frontal lobes of the brain are involved in personality and impulse control, but we can't be fully confident in that claim, not based on the case study alone anyway, because we can't rule out other possible explanations for why he changed. So, yeah, perhaps it was the brain damage that caused him to behave differently, but it could have also been the sheer stress, the sheer trauma of going through such a gruesome experience, right? Or it could have been because the accident left him disfigured. So maybe that made other people perceive him differently and treat him differently. And that could also explain why he behaved differently after the accident than he did before. So, case studies are useful because they often represent the only way to study a particular phenomenon, like human brain damage. And like anecdotes, they can inspire hypotheses that can be tested using other methods, such as experiments involving laboratory animals. But they too are limited in that they don't allow us to generalize or to infer causality. Now, we can address at least one of those limitations of case studies by conducting what your textbook refers to as descriptive studies. So, for example, let's say that I'm interested in a variable like frequency of sexual intercourse among married people and among single people. So, if I want to know whether those two populations differ in terms of how much sex they have, then I could administer surveys, right? Where I ask some married people and some single people to report how often they have sex. Now, obviously, I'm not going to collect data on every individual who belongs to both of those populations because those are huge populations, right? So I'm just not going to have the time or the money to be able to do that. But as long as I ask my questions to enough people from each population, as long as my sample size is big enough, and as long as I'm selecting people for that sample randomly, then I'll be able to be fairly confident that my samples of single people and married people are representative of the populations from which I drew them. And that way I can generalize. I can take what these single and married people told me on my survey, and I can use it to make an inference about the larger populations of all single and married people. So if I find that the married people in my sample reported significantly more sexual intercourse than the single people in my sample did, then I can be reasonably confident that married people as a whole, that married people in general, tend to have more sex than single people do. And of course, the same could be said about any correlations that my research discovers. So, for example, if I find that people who score highly in job satisfaction also tend to report higher frequencies of sexual intercourse, then I can conclude that those two variables, job satisfaction and frequency of sexual intercourse, are probably also correlated in the population. So, to some extent, descriptive studies do allow us to make generalizations from the smaller groups that we collect our data on to the larger groups that we're actually interested in. However, like case studies, descriptive studies are also limited in their abilities to draw conclusions about cause and effect relationships. So, for example, you're probably familiar, I hope you're familiar, with the saying that correlation does not imply causation, right? And it doesn't. Just because two variables are correlated with each other, that doesn't necessarily mean that one causes the other. So, if it's true that job satisfaction and frequency of sexual intercourse are positively correlated, then that would be an interesting finding. It's one that would allow us to predict with better than chance accuracy how much sex people have based on how satisfied they are with their jobs, or vice versa. But it doesn't necessarily mean that high job satisfaction causes high frequency of sexual intercourse. After all, the relationship could go the other way, with high levels of sexual intercourse somehow causing high levels of job satisfaction. Or it could also be that each variable is correlated with a third variable, and that the correlation between job satisfaction and sexual intercourse just comes about as a byproduct of those other separate relationships. For example, it could be that happy people tend to have more sex, and that happy people also tend to be more satisfied in their jobs. So, in other words, maybe it's happiness that causes sexual intercourse to increase, and maybe it's also happiness that causes job satisfaction to increase. So, in that case, there wouldn't be a cause and effect relationship between sexual intercourse and job satisfaction in either direction. Those two variables would be correlated with each other simply because they each have their own separate correlations with this third variable, happiness. So, whereas we can use descriptive studies to describe a larger group of people or animals on the basis of a smaller group, like a sample, we can't use those descriptive studies to infer causality. We can't use them to say anything about cause and effect. And if we want the evidence that we collect to tell us something about cause and effect, then we have to conduct an experiment, which is the last type of research method that the textbook discusses. And I think this will probably be mostly review for you. I'm sure you've learned about and read about experiments before in other classes. And the textbook describes several different types of experiments, but what they all have in common is that they always include at least one independent variable and at least one dependent variable. Now, remember that experiments are designed in such a way as to provide us with evidence about cause and effect relationships. So, when we conduct experiments, we manipulate the independent variable. We make that variable change, such as by exposing some participants to one level of it and other participants to a different level of it. And then later, we measure the dependent variable. So we manipulate the independent variable and we measure the dependent variable. And of course, we do this because we hypothesize that by manipulating the independent variable, we're going to cause changes in the dependent variable. So we think that the measurements we get on the dependent variable are going to differ as a function of the independent variable. So, guys, a good way to think about the independent variable is that it's the hypothesized cause. Okay, it's what you think is going to cause a change in something else, and that something else is the dependent variable. So you can think of the dependent variable as the hypothesized effect. Now, I also think it's helpful to explain why we refer to these variables with the terms independent and dependent. Guys, we use the term independent variable because we want this variable to be the only thing that differs. We want it to be the only thing that varies across the groups or across the conditions that we're using in our experiment. We want that variable to vary independently of everything else. We don't want anything else to vary with this independent variable. So the effects of every other variable are held constant or balanced out across our groups or across our conditions. So we're isolating the independent variable as the only systematic difference in how participants are treated in the different groups or in the different conditions. And that way, if those groups or conditions differ later on the dependent variable, then we can point to this one difference between them and say, that's why. The only way they were different earlier was that they got different levels of this independent variable. So if they're different now on the dependent variable, then that independent variable is the only thing that can account for that difference. So remember, the independent variable in an experiment is the hypothesized cause. It's the variable that we manipulate in an experiment. The dependent variable, again, is the hypothesized effect. It's the variable that we measure in an experiment. And we call it the dependent variable because we think that the measurements we make there are going to depend, at least to some extent, on which level of the independent variable a participant experienced before we measured them. So, for example, if you're manipulating the amount of sunlight that different plants receive, and then you're measuring the growth of those plants, you might hypothesize that the amount of growth you see in a particular plant will depend on whether it got exposed to a lot of sunlight or a little sunlight. So the independent variable in that case is how much sunlight the plants were exposed to, and you're predicting that the different levels of that sunlight variable will cause differences in the growth variable, which is the dependent variable in this case. Okay? So please let me know if you have any questions about independent variables and dependent variables. But now that we've reviewed those, let's move on and talk about the different types of experiments that a learning researcher could conduct. Now, the basic distinction here is that there are between subjects experiments, what we call between subjects experiments, and then there are what we call within subjects experiments. Okay, so you've got between subjects experiments and within subjects experiments. And I'll talk about between subjects experiments first. Guys, in a between subjects experiment, what the researchers do is they take their sample of participants, they take the people who have agreed to participate in the experiment, and they divide those people into at least two groups. We call those groups the experimental group and the control group. Now, a between subjects experiment can involve more than two groups. For example, you can have more than one experimental group alongside your control group. But for the sake of simplicity, I'm going to focus here on between subjects experiments that only involve two groups. So, like I said before, we want to design our experiment so that the only difference between these groups is that one group receives one level of the independent variable and the other group receives the other level of the independent variable. So we want to begin our between subjects experiment with two groups of participants who are equivalent, two groups that don't have any pre-existing systematic differences between them. And then we're basically going to do something with the experimental group that we don't do with the control group. Now, this concept of starting our experiment with equivalent groups, we've got a name for that. We call it the principle of initial equivalence. Okay, the principle of initial equivalence. And this is a very important principle. So we do want these groups to be equivalent initially. We want to start our experiment with these equivalent groups of participants because then we can make those groups different in one way and know that that's the only systematic difference between them. We want to start them off the same so that we can make them different in one and only one way. So, the question becomes, how do we satisfy that principle of initial equivalence? How do we start our two groups off as equivalent to one another? Well, there are a couple of different ways that we could go about doing it, but the most common one is to use a technique called random assignment. Okay? We call it random assignment. So we need to assign each participant to the control group or the experimental group, meaning that they don't pick which group they want to be in. We put them in their groups, right? And we want to make those assignments completely at random. So, in other words, each participant should have an equal chance of winding up in the control group or in the experimental group. So, for example, one way we could do that would be by just flipping a coin to determine which group each participant is going to belong to. So that way, each participant would have the same chance. They'd have the same 50-50 chance of being assigned to one group or the other. So that's how random assignment works. And again, the reason we use this process is because it helps ensure that as long as our sample of participants is big enough, then the differences that exist between individual participants will get sort of evened out across our two groups. For example, individuals differ in a lot of ways, right? Some are smarter than others, some are more extroverted than others, some people are more motivated than others. But what random assignment does is it tends to balance out those individual differences so that we don't wind up with more intelligence, more extroversion, more motivation, or more anything else overall in one of the groups than we do in the other. And that way, if we do observe a difference between the groups on the dependent variable, then we'll know that that difference was not due to a pre existing difference between the groups in terms of intelligence or extroversion or anything else. No, that difference on the dependent variable could only have been caused by the difference that we introduced on the independent variable. So, again, the reason that we use random assignment in between subjects experiments is to help ensure that the only systematic difference between the control group and the experimental group is the difference that we created by manipulating the independent variable. Okay? Now, this concept of using random assignment to satisfy the principle of initial equivalence is a really important one. So, to help clarify how it works, I want to show you a little video now where some researchers are going to talk about how and why they used random assignment in an experiment they did to investigate the effects of violent video games on aggressive behavior. So, here's that video.
The independent variable in the experimental study was expected to be related to the dependent variable in the following way. We expected those who had been randomly assigned to play the violent game would later on behave more aggressively than those who had been assigned to play the nonviolent game. And for that reason, we want the control group to be essentially the same as the experimental group in all features, as many as possible, with the exception of the independent variable. As we want them to be the same kind of people, we want them to be treated the same way by the experimenters, we want them to play a game that is as equally enjoyable or frustrating. We simply want the groups to differ on one thing, and that one thing in this case is, does the game contain violent content or not?
One way the researchers achieve this sameness between groups is through random assignment of the subjects.
In order to show cause and effect relationships, one needs to assume that the groups in a study, the participants in a study, initially began as equal or nearly equivalent. The best way to ensure that is to use some kind of a scheme where individuals who have agreed to participate in the study are assigned to an experimental or control group based on chance. In this way, the characteristics of individuals that differ naturally in the world, that these characteristics could somehow be evened out across experimental and control groups initially.
What we do is randomly assign participants to either the control group or the experimental group, and random assignment means that each student had an equal chance of being in one of those two groups.
Random assignment is essential to valid results. For instance, if Anderson and Dill had allowed the players to pick which game to play instead of assigning them to games, the more aggressive players might have chosen the more aggressive game. It would have been impossible to determine if any later aggressive behavior was due to the game or to just their personality.
Okay, so random assignment is one way to satisfy that principle of initial equivalence and ensure that the only difference, the only systematic difference between the control group and the experimental group is that the folks in one group get one level of the independent variable, like a violent video game, and the folks in the other group get a different level of the independent variable, like a nonviolent video game. Again, the logic of a between subjects experiment relies on this assumption that the groups begin the experiment as equivalent to one another. Now, there are other ways to satisfy the principle of initial equivalence. For example, the textbook talks a little bit about something called matched sampling, or what's also known as a matched pairs design. So that's another way to do it. But simple random assignment is the most common way to start out with equivalent groups, and it usually works as long as the groups are big enough. So that's why we use random assignment in between subjects experiments. So remember, in a between subjects experiment, you're manipulating the independent variable by exposing each participant to one level of that variable or a different level of it. So, for example, in that experiment on video games and aggression, each participant would play a violent video game or a non-violent game, and then they'd have their aggression measured afterward. Or if you were doing a between subjects experiment on sugar and hyperactivity, then you'd probably have each person consume something with sugar in it or something without sugar in it, some kind of placebo that didn't contain sugar, and then you'd measure how hyperactive those people were. So again, in a between subjects experiment, each participant gets exposed to one level of the independent variable or another level of it. By contrast, in a within subjects experiment, each participant receives both levels of the independent variable. So if you were going to take part in a within subjects experiment on video games and aggression, then you'd first play the violent game and have your aggressive behavior measured. And then, sometime later, you'd return to the lab, you'd play the non-violent video game, and then you'd have your aggressive behavior measured again. And of course, other participants would go through the same basic procedure, but for some of them, the order would be reversed, meaning they'd play the non-violent game first and then the violent game later. So, in a within subjects experiment, it's kind of like you're participating in both the experimental group and the control group, just at different times. And there are advantages and disadvantages to this kind of design. So, for example, one nice thing about within subjects experiments is that you don't need as many participants for them as you would for a between subjects experiment. After all, if my goal is to get 30 measurements, 30 scores of aggressive behavior after each type of video game, then if I'm using a within subjects design where each participant plays both games and has their aggression measured after each one, then I'm only going to need 30 participants, right? As opposed to the 60 that I'd need if I were using that one or the other between subjects type of design. So that's one advantage of within subjects experiments. And another one is that with them, you don't really have to worry at all about whether there are any pre existing differences among the people who are getting the different levels of your independent variable, because in a within subjects experiment, you've got the same people participating in both of those conditions, right? And so those are nice perks of doing a within subjects experiment. However, a drawback to this kind of design is that it does introduce the possibility of order effects where participants might behave differently in the two conditions, just depending on which one they experienced first. And another related problem is that a within subjects design can also make it easier for the participants to figure out what the hypothesis is. So, for example, if you play both types of video games, the violent one and the non-violent one, and then you have your aggressive behavior measured after each one, then you might infer correctly that the experimenters are expecting you to show more aggression after the violent game. And so, if that's the case, then your knowledge or your suspicion of how you're expected to behave could wind up influencing your actual behavior. So, depending on what it is that the researchers are studying, they may decide that a between subjects experiment or a within subjects experiment is the more appropriate way for them to test their hypotheses. Okay, well, guys, one more thing that I should mention about within subjects experiments before we move on from them is that your textbook also goes into some detail about a particular type of within subjects experiment, which is called an ABA reversal design. We call it an ABA reversal design. Now, the A in this type of experiment represents what we call the baseline period. Okay. And during the baseline period, each participant's behavior is observed before the experimenter has exposed that participant to any sort of treatment, any sort of intervention. Then the participant's behavior is again observed while the treatment is in effect. So the B in an ABA reversal design represents what we would call the treatment period or the experimental period. And then finally, we'd implement one more baseline period, one more A period, to see if any behavioral changes that occurred during that treatment period persist or whether they go away once the treatment has been terminated. For example, let's say that I'm interested in whether hot temperatures have the effect of reducing motor activity in rats. And let's say that I think they do. I think that hot temperatures cause reduced movement, reduced motor activity in rats. Let's say that's my hypothesis. So, to test that hypothesis with an ABA reversal design, what I do first is observe how active those rats were at normal room temperature, right? So that first observation period would just be a baseline period. It would correspond to the first A in the ABA reversal design. I just want to see how active the rats are under normal circumstances. How much do they move around before I've manipulated the temperature? And then, after I've observed the rats at baseline for a few minutes, I'd crank the temperature up, right? I'd expose them to a treatment period. I'd expose them to the B period in the ABA reversal design. And if I observe during that period that they become less active than they were during the baseline period, then that's consistent with my hypothesis since I predicted that hot temperatures would reduce their motor activity. But I wouldn't necessarily know whether it was the heat itself that was causing that reduction or whether it was something else, like, say, fatigue, that caused their activity to decrease. So, to rule out that possibility, I'd reinstate the baseline condition. I'd return the temperature to normal. I'd impose one last A period, and I'd see if the rat's activity levels went back to normal. And if they did, then I'd have some pretty compelling evidence that it was, in fact, the hot temperature during the treatment period, during the B period, that caused their activity levels to decrease. Okay, so again, the ABA reversal design is just one specific type of a within subjects experiment. Okay, well, that's about all I have to tell you about those sources of data that are described in the textbook. But as always, if you have any questions about this stuff, then please let me know. Take care.